I will give a talk on laser influence on biopolymer media with carbon nanoparticles for biomedical application. As you know, there are many types of carbon nanoparticles. Some of the most popular are graphene sheets and twisted graphene sheets into nanotubes. Materials based on these nanoparticles have interesting electrical, thermal, mechanical, and optical properties. In my report, I will talk about materials with a nanocarbon framework made of carbon nanotubes. Materials based on ordered and disordered systems of nanotubes are used to create flexible electronics and bioelectronic components. For example, that could be attachable and implantable electrodes for neurostimulation, electrocardiography, electroencephalography, temperature, pressure, strain sensors, biochips for detecting infectious diseases, and others. Framework nanomaterials based on ordered and disordered systems of nanotubes in biocompatible matrices are widely used to create bone, cartilage, and heart implants containing drugs and cells of the corresponding tissues. So why can't nanotubes be used to create tissue engineering matrix? Nanotubes have a size similar to the size of the main component of natural extracellular matrix. Their mechanical properties are similar to proteins of the extracellular matrix, and adding carbon nanotubes in materials of implants increases adhesion of cells, their proliferation and differentiation. Currently, laser radiation is widely used to synthesize nanomaterials, including carbon nanotubes. So, because laser irradiation provides non-contact localized thermal zones with high-speed heating. So this leads to improved mechanical and electrophysical properties of nanomaterials based on nanotube networks. Surface of carbon nanotubes contains multiple defects. Defects are formed as a result of the nanotube synthesis. Most often there are types of defects, one vacancy of carbon atoms, two vacancies, and stone wells defects and their combinations. It's known that carbon nanotubes can be welded by laser irradiation. How can we form X and T-shaped connections? So when the laser radiation is directed at nanotubes with defects, the defective areas will be heated more strongly, as the thermal conductivity in these areas is much lower. Therefore, the most likely areas of the spliced tubes are defective areas of tubes. So this slide shows the simulation of the laser bonding of two nanotubes heated by laser. We also carried out numerical simulations of the carbon bonds formation with different types of defects. From the enthalpy graphs, it's shown that the most profitable energy process is the formation of a nanotube framework with several combined defects. In this case, the largest number of chemical bonds between the tubes is formed, such as connection thermodynamically more stable and mechanically strong. The next slide shows the character of changes in the number of covalent bonds between the nanotubes during laser irradiation. The time dependencies at various distances between the tubes from 3 to 3 and 7 angstrom are showed. The next slide shows, in experiment we create a nanotube welding area using a pulsed laser. Using mathematical modeling and electron microscopy investigation, we determined that intensity of radiation for welding nanotubes together. Same images, scanning electron microscopy images demonstrate the accumulation of electric charge in the region of the nanotube juncles, so it can be seen by the light areas on pictures. It's due to the low electrical conductivity of imperfect amorphous carbon junctions. We can see such an effect in crystalline junctions. Based on the research, we proposed a technology for forming networks of nanotubes in scanning mode to create planar frame nanomaterials in a given area. From nanotubes and hybrids of nanotubes and graphene, such materials can be used to create nanowires in integrated circuits, for example. But to create biocompatible 3D materials, we need to form a framework of nanotubes in biological metrics. We have chosen the proteins albumin and collagen and the amino sugar chitosan. Next, we made homogeneous aqueous dispersion media with these biopolymers and with nanotubes. This slide shows how we create our biopolymer nanomaterials. First step, we applied layer by layer to the substrate. Second step, we transform dispersion by laser radiation into solid state. This layer by once again layer by layer. The result is a material with carbon nanoframework. On the slide, you can see that molecular model of such nanomaterial. The mechanism of laser radiation interaction with carbon nanotubes in a liquid is as follows. First, there is absorption of laser radiation. Then the laser energy is transforming to heat, and then there is a formation of bubbles around the tubes. As a result, porous material is created. It has been established that the change in the porosity of biopolymer framework nanomaterials depends on the radiation energy density. An increase in the radiation intensity above the threshold value affects the increase in the porosity of nanomaterials in the matrix of three biopolymers. We study morphology and porosity of structures in more detail by scanning electron microscopy. The structures have a bimodal pore size distribution, as it can be seen from the slide, from 1 to 5 microns to provide the neoinnervation and neovascularization process, and from 100 to 200 microns for adhesion and proliferation of cells. Nanomaterials have a porosity of about 70%. We also controlled the surface structure of nanomaterials. When we change the process of creation of biopolymer nanomaterials, we can vary its surface, for example, to interact nanomaterials with the different cell types and tissues. The connection of the oxygen atoms to nanotubes leads to tube deformation. The more oxygen atoms, the stronger is the deformation. Nanotubes configuration can be changed by changing the points of the oxygen tube connection. This can be used in the production of biopolymer nanomaterials with a certain internal structure. The formation of nanotubes frameworks in biopolymer matrices provides a change in mechanical characteristics. It has been proven that the hardness of nanomaterials obtained by laser are significantly higher than the hardness of materials obtained by thermal heating. The hardness of laser nanomaterials is in albumin matrix reached up to 480 megapascals. The nanomaterials are electrically conductive due to the nanocarbon network. We compare the conductivity of nanomaterials created by thermostat, and by laser. As a result, nanomaterials created by laser had the highest electrical conductivity. This is very important because cardiac implants must conduct by currents. Thus, the structural and electrical properties of biopolymer nanomaterials are ready for placing cells in them. This is necessary to create cells patches that will be implanted on the area of myocardial infarction. First, we studied the interaction between carbon nanotubes and human fibroblasts. On the slide, you can see best adhesion of cells and their lateral processes to the surface of nanotubes in comparison with a smooth surface without nanotubes. Cardiovascular implants must be compatible with the endothelial cells, and the best cells viability was demonstrated on the albumin layers after 48 hours from start of incubation. And using MTT assay and fluorescence microscopy, it was shown. As we can see from the images, cells accumulate in groups. The close arrangement of cells in groups provides to the formation of tissue monolayer. It can be seen that cells repeat the porous structure of the composite layers. To improve self-proliferation in the structure of nanomaterials, we have developed a device for electrical stimulation. The device consists of a pulse generator, printed circuit board on a culture plate, and electrodes in contact with which contact the biopolymer nanomaterial. Electrical stimulation of fibroblasts on laser-formed nanomaterials provided better cell growth. This was due to the control of the transmembrane potential. Also, electrical stimulation provided an increase in the cardiomyocytes growth ratio. Since electrical stimulation of cardiomyocytes is able to provide the synchronous beating instead of spontaneous. Thus, we obtained the filling of the porous structure of biopolymer nanomaterials of cardiac cells. Before experiments in vivo, we carried out studies of the gamma compatibility of nanomaterials. We investigated blood hemolysis after its contact with nanomaterials. The value of hemolysis was no more than 1% for nanomaterials based on 3 types of biopolymers. It was albumin, collagen, and chitosan. We implanted biopolymer materials into the infarction area of laboratory animals, and it was observed that deviations in the blood analysis and the cardiogram. But after from 3 to 4 months, these deviations decreased. So as a result, using histological and immunohistochemical studies, we determined that the biopolymer nanomaterial was bioresorbed after 3-4 months, and during this time, myocardial recovery was about 70%.